Everybody have a good night's sleep yesterday? Yes. The power didn't go off, so it was actually very comfortable. And I had a pretty good night's sleep, so I have a lot of energy. So be ready to have a really good day today and continue our days of high energy and high productivity and have a lot of fun.
A lot of sessions on the Nichipu Kuchi, that is Zulangesi Hotel, Sanity, Zendratia, Nishim Ben, Shudo, Sanitiranga Sushi, good port in the DJ, any two Gani, any Zendran trains.
Before we do start, I do want to say one thing that really touched me yesterday. A couple of you invited me to go play soccer yesterday. Oh, see, it's not working. Give me 1 second. Troubleshooting. There we go. So, I was invited to play soccer yesterday, and I have to say, I haven't played soccer since I was 15 or 16, and just spending time with you and having such a great time, it really touched me, and I want to thank you for such a great experience.
Ani kesan chetu khachiga council policy ga ke tansons to stay by yes the pay two chansons tangara imbana council polo de da lu chunga in caps jela ya ta tanda pa da simanyos ro what is tambala pay two chansons sama to chase.
As you may have seen, I'm not very good, but I had a lot of fun. We laughed a lot, we bumped into each other, we fell, I scored a penalty kick, and I really had a lot of fun.
That's to Toyo Sanga Tina, say I will marry Simba in Aya, Tangasuchi, Nyam de Kemugere, Simus the Tachi Desha Tangare, Saladria, La Sopachi, the Kesan Shutu Johnsons.
And being part of your culture and really learning about your culture has really touched me and inspired me. So I'm going to try to teach with this on, which I bought yesterday. Thank you to our friend that took me to Camp 3 and he helped me find this. Apparently it's from Mongolia, I believe. It's super heavy and it's hot, but I'm going to try to use it.
Tanda banangi chesugo revision ko korte dus jangage thapsite tang yostin stamba tirangin zendrate trip kapsa de shamudi konchi chayis shamudi kesanga so the game sumbalan rukeps ani rokpachi ke rokpachi che new bayens ta shamudi pe de ta yada nga so de sopo ge shamu sares tako jikoso shuchdu ta sawasabe kas kona sawshuchdu simbana ya konchin zendrate chayis.
So maybe it'll last a minute, maybe 5 minutes. We'll see. Why is this not working? Well, we can just do it through here. That's fine. So I do want to spend some time. Chris will also talk for a few minutes on our Mori points from the last couple of days. I noticed that this was added on the day 1 Mori point, so I'm just going to review it very quickly.
Handa tadanga so tokyo ge twa khachi orwa te ga stagin di parola ya gen Chris ge yan de parola ya se chi go chi gres ta de jate ko ge nyima tampo ge te gang la nyima tampo te ga trisha sim bai na te da len ja ma chong da te de ja gens.
So somebody asked if every single cell of our body has the ability to reproduce, how about cells of our hand? That's what I'm gathering for this. Is it possible for if one of the man hand cell put into the egg and then becomes a baby? So basically you're asking if a cell from the hand can potentially reproduce and be used to create a baby. And that's what I'm gathering from the question.
Zupugi tapon re la ya korang chipechia gi nyubat yo pa imbai na sro ane nga suga lakpa gang ge tapon tilaya kare chongre sen nga suga lakpa mi chi ke lakpa gang ge tapon chi len de na tapon ti ani khamer tapon to nyam de ko dijur chi gi pa imbai na tilaten de tanga suga bugi chi zut kroe zut na kanda zut kres.
So I think the best way to tackle this is by saying the following. There are different kinds of cells, just like there are different ways for cells to divide and reproduce.
하다가 가지고 타폰이 바이나 타폰코 가서 그래. 그런 자귀지에다 아니 지페지에기 탑블 만들어 바나진 타폰라 임바이나야 타폰라야 지금 만들어 요레스.
So just like we learned yesterday with, or not yesterday, but 2 days ago with mitosis, this is a process which creates two identical cells from one mother cell.
하다가 수님이 이거를 가지고 될라 따라와 주폰지기 나바데 따라와 주폰지기 나바데 가레 시스템발야 타폰지 자구치 되던 타폰지 버치찬이 듣고요와.
And you may recall, we haven't touched on this subject yet, but you may recall we also have meiosis, which is different from mitosis.
당가 속에 조시디 트리오 말이었어. 임바나 시치나 치수 담바수 그래서 가 속에 제폰 시지세 줘라. 제폰 시지세게 타폰 차구지 남바지 그래서 데스 폰지단 로아레스.
It is through the process of meiosis that we get the emergence of gametes, which are sex cells, both sperm and eggs.
다 단단가 속에 제폰 시지게 남바텔라테네 데가 속에 데 감두티 종구요레스 감두텔라야 가서 감카르멘샤 종구요레스.
And you may recall, if you have a cell with a full genomic count and it goes through meiosis, that count is cut in half. So it goes from 2n, being a complete genomic count, to haploid, 1n.
다 단다 최초게 제폰 시지게 남바티 땡게요 바인바나 담봉아서 요약가서레세나 타폰 시그나나 요 비기 르지기 답차테코 다 2n 담니니 요로레 마레 다 띵아서게 제폰 시지게 남반 루알 텐데데 깡치 차고레스 다 다당아서 작은 타폰데 약간 타폰 차고요레스 단데 약간 타폰데 가서 감득나나 요로와.
So in order for fertilization to occur, we need to have two gametes, a sperm and an egg, fused together, have the process of fertilization where we go from 1n plus 1n equals a cell, which is a zygote, with 2n, a complete genomic count.
단다데 값승아수 다고 양치 다 감드들의 양치가수 그 타폰지 접어야라야 감드 임바이나 감드 그나라야 릭제테코 다가수 약간 지대요 와티 냠드 트레랑가수 감감런션 지데티 아니 시조치와라 사분 시조치와라 테네데데 가수 타폰나라야 양치 릭지기 땅가테 2n 로다 담지 차고로 차차지야 차고로 딘다티와라 테네데데 가수 그다 사분 디공 그 타폰시 듣고요레스.
We clear on this question? Thank you. And the second question that I was going to tackle before we continue, which was also added on day one, somebody asked, can we create new cells?
So
this is a question that is actually quite complicated, and through technology, through all kinds of research, this has allowed us to study and understand the evolution of life and the emergence of the first cells.
단다데게 좋아데게 다고 렌티 군욕부여레스 다 저기 만나 단다가 주 트리기티 열진 치발라텐데데 가수게 다 세속게 벤주다 아니 차폰 토마데 간도 종레사 저거와 대강라야 아니 고다 딱세치야당 고토 펠라다 열지 두고요레스.
We can, for example, theorize on how the first cells came to be, how they were created in on our ancient planet, billions of years ago.
달로 담바 망보고라야 아니 차폰 토마티스 그러다가 속에 사대강라 간다도 지종메 메텔라야 남사사고요레스.
We can also ask, can we, in modern time, create new cells? And there is research called synthetic biology, which allows us to create pseudocells in a lab.
단다게 드와데 카레스나 다 ngo마티 마임바 단다게 티캅텔라 가츠가 타폰 서와치가 주트 크르베세티 오레스 다 단다 임바나 가츠게 synthetic biologist na jotrub gi chingu pati na laya ni chingu ge takshe kha laya ani thapon da chanda wa tinda ne khusu zutun chi yores.
So the topic of artificial creation, especially in places like California, the Institute of Technology of California and MIT, Massachusetts, has actually done a lot of work with the creation of synthetic cells in a lab setting.
다 단다게 지 피게 다 랍다 첨부 MIT 게 고게 탁세 칸라 소파 탁세 카치가나라야 아니 단다게 고 유튜브 지와라테네 아니 탑폰 차다와서고 왔다 시도 시스템발에 오늘 줌마서고 다 찬다운도데 조야주가 조야기 탁세 수 첨부 지기 요레스.
But do remember, just like we learned from Chris in our lectures, in nature, all cells come from other cells.
다 겐 크리스긴 전단하는 가수 장반나 신시샤바인바나 가리 지고레스나 다 랑숑궁우니 지바인바나 차폰 참마 차폰 젠리 종고 요레스.
Good? Wonderful. Thank you. Would you like to?
Yeah.
Chris also has a muddy point he'd like to cover for a few minutes, so let's pay close attention and listen to him.
다 겐 크리스기 아니데 돈드치라 레렌치자그레스 티기 질랑가 순전드메트기엔스.
That was 5 minutes with the hat.
It's very heavy.
I just wanted to touch base and clear up what I think were some misconceptions about the cells that we drew yesterday.
카세즈야 카지에스도 카세즈야 계상아수 타폰골 개지셰버와 되게 나라야 지금 구안노알렌주 카치두스티 바디 카세즈기엔스.
First, this is the model that we did is mostly for single-celled organisms. For human cells and mammals and reptiles, it's a little bit different.
하다다 계상아수게 지금 베이직 조조데 만치신바나 타폰치게된게 징글레 시샤데 땅다 지주요레스 땅아수 네스 소차레 미레 디나징게 게르징게 소차 디나도라 타바인바이나로와 아니 this 만도요레스.
In humans, glucose is in your bloodstream and enters your cells and is not processed by lysosomes.
다 피샤샤바이나 가수게 karsa mila chombaimana ta gulungarji chiwara gulungarji te ngasun royala kandasun dogore ngasu thlam ne thapongla dogorwa thapongla dustambala ko shishusuni tin jezguti maris lyzosum te maris.
And in single-celled organisms, food is broken down, and then there are many more processes to prepare that food as glucose for mitochondria. There are many other steps.
Can you repeat that again? Sorry.
In protists, if like food enters the cell, it gets broken down, and then there are many other steps to prepare that food to be glucose.
In a single cell.
Yes, in a single-celled organism.
Okay.
So like as food gets broken down by the lysosome, but then there are many other steps before that food is ready to enter it's turned into glucose so that it can be used by the mitochondria.
Okay.
Does that make sense?
Yeah.
Okay.
The Tapon Chidingi Chungitinado in Bana, Koganala, Sama Chilangroa, Sama Chilan Stambala, Samata, Jesuku, Shishuzugora, Lajusum Groa, Shishuzu attended that day, that the Tamatilaya, Nuba Jurdating, and Sugunichi Tapon, Nuba, the Peshutango, and Abachichagora, the Charo Porto Kolaya, and it's a Jerem, not for Mampuchi Juguris.
And the last thing I wanted to clear up is even though the Golgi apparatus does move most things throughout the cell, ATP is actually moved by a slightly different process than the Golgi apparatus.
Nubati ATP kungusha jatante gozgi latene maimba kungusha jata mandawachigores.
So overall, everybody understood the right concepts and had the right organelles doing the right function, but there are a couple of small details that I wanted to make sure you all were aware of.
Ahada kesangasu chekudripikasuma cheshigete yapana shasta ko samachilanchi ti nubala jurchi da ko pechutango yopana so chizugo yorwate zuage ani wanthati kanza pechutanga zuka tapado chesha simbana dela yajian ko shepthagi yon Thank you.
So I'm having some issues here. Give me 1 second, please. Let me just deal with. Take a 1 minute break.
Come on, chicken person science.
While I was doing that, I was looking at your all of your petri dishes, You all have some very wonderful stuff growing, so good job swabbing gross places.
Ta kesa nga su de la ya chi dharmach kana tele ya ma ka sa ta chingwi ting nga su ta demjing do su ko wa ta this Istanbul tangi chita ba yin stin ana pe shiu chi ya chi yong do stin Istanbul che su pe te sachen zon zo chola ya so te ta nga su ge ma pi te len chi na chi yor wa de ka pe ap drip chas pe ap chi yong do sa.
Okay, I think it's working now. We'll give it a try. So for today, the topic of the day is going to be central dogma.
Ta tire nga su jo shite ka re se na tawa nyimpu ins.
And of course, central dogma deals with the change of energy, of the interaction of energy going from DNA to RNA to protein.
Da tawa nyimpu de ke kapsa nga su kare ke jurum chinde se ja gore se na DNA ni RNA la juro RNA ni ane tzi la juro ge jurum tinde se ja gore.
And of course, proteins make up genes, and when they're expressed, they are manifested as some type of manifestation and physical property within our bodies.
Ta tanda nga su ge tzi se te ane tzi se te tires no rigzila tene chongo res. Ta ti nga su rigzila tene chongo yo cham la ya khu ge ta chi ga namba ngoya te nga su ge zukpu ge chechu ge namba ya ngong res.
So for our objectives for today, the focus, of course, is going to be the central dogma going from DNA to RNA to protein. We're going to do a few things. We're going to spend a few minutes reviewing DNA and RNA structures.
Ta tanda nga su ge jo so de tawa nyimpu che nga su DNA ne RNA ne tzi la trebe ge namba tires. Ta ti ge no ne tampu te la nga su ge DNA ta RNA ge chakola ya nga su cha che ins kama khashi ji pe chi tang.
We're going to identify the steps of the central dogma, which is, of course, this particular flow of life.
Ta ti ge je la ya nga su ge tawa nyimpu na na yo pi ge ko ge jirum chi go ro korum chi go ro tela ya ta ge ins ta ti ge korum te chir ge na phazu te la rumu chi tsha dwa o ti ge jirum tin du yo res.
After our first break, we're going to start in our activity where you actually get to apply the central dogma.
Ta tanda nga su socha person ge je la ya nga su tawa nyimpu ti pecho tanga yu chu ge ani che shegu chi de ins.
We're going to be using coded messages and applying transcription and translation to find hidden messages.
Ta tanda nga su kare chigre se tambala ya tanda ani sangda tina pecho tang de de wa sangda ti pecho tang de te ani tela ten de te nga su ge phapshi tang phajur ge juram ni sha do de te thama tela ya sangda tina ne ani da kare tek du se te karim ba chogi yes.
And just so you know, this happens to be one of my favorite talks that I always give in my lectures, in my biology classes.
Ta ngara chingi chari trebi kapsu tanda ge joshi te ne ngara joshi gashi te yes.
And this particular activity was recently published, and all kinds of universities use it with their students. So I'll be using it again with our class today.
Ta tanda ge ning ngasu chotan jiba dia ge chegu te imbaina ani chegu te niche ta de ko pattern chi sha ju ge chegu chire te chegu pattern chi wala te de te ani ngarang ge jada nanel hotla phala peche ta ba yen te bete sulo hota chambo mampu na na dus peche tang go yores this tambala ya te tira ngasu ge dwala ya ani peche tang yes.
We're also going to learn about the importance of why there are certain sequences of DNA that have to remain conserved throughout all kinds of organisms.
Ta tem se ne chingwi na chope ge na la ya nga su ge khu ge kar su gre ta DNA ge khörim chi go wa khörim te katti ba na tembu de gu yo pa nyer so chi gu yo pa ti ge ke ching ran je te on de se ja ge yes.
We're also going to learn about mutations. We'll start getting our understanding of mutations, which I believe Chris will talk about more tomorrow, if I'm not mistaken.
Yes.
So we are going to start our basic understanding of mutations.
Da te je la ya nga chu ge ninjur yo ra ninjur go ko la ya ne ngutu te chi chi ge yes ta ninjur go la ge janta ngu shi ge ta je bo ta te sanye ge Chris la ti res.
So in our activity, we're going to use all kinds of themes to really get an understanding of the importance of the central dogma and what it means for conservation of sequences and how that relates to mutations.
다 단다가 제구티 드립데데 가르치긴 시스템 방아 속에 타와님부 지거로 왔다 징이 다 세속의 타와님부 되게 드럼시까지 임바체 되게 나네 네 거기에 DNA 기 거기 리지기 코름티 녀석지 그하게 게네랑진 가림바치 가르치는 게 첨부리스로 아니 딜라텐데데 데탈라텐데데 아니 되게 가속에 가서 버리다 닌줄라야 슈신 가르쳐 그래서 지거로 데탈란두스타기인스
Good? Excellent. So let's begin. We're going to start talking about, this is very sensitive. There we go. So we're going to start our fundamentals today by reviewing the distinctions and similarities between RNA and DNA.
다 당보들랑 가속에 DNA 당 RNA 인식이 바로라야 손착가리도 아니 체벌가리도 됐다긴스
So within your group, for about 3 to 4 minutes, just look at these drawings. We have DNA on the left, RNA on the right. Understand how they're similar and how they're different. And come up with a list within your groups of similarities and differences.
다 단다 수수수로 커너네 그러면 숨쉬시그린라야 아니 파조테 DNA지 저조테 RNA로와 DNA 당 RNA 나나 아니 손착가리도스 And
after we're done reviewing that in your group, we're going to list them on our board. So start discussing it within your group. Spend about 3 to 4 minutes. I'll let you know when we're done, and we'll start drawing them on the board. Do we have an eraser for this? Oh, here we go. Thank you. Dogma, as they're giving their answers, and I write them in English, can you do the translation in Tibetan? Thank you. So that way they can see. One more minute. We're good? All right.
That's all.
So, any of the groups want to tell us one similarity between DNA and RNA?
Aha, rukachi ki DNA ta RNA yu paru ko choncha ching de sijar chidas. Yeah, they both of them consist of nucleotides.
They use nucleotides, right?
Yeah.
So use nucleotides, good. Good. Another group, another similarity.
Aha, ruka jenda chike yang ko choncha jenda chilaro chidasa.
Nicola adenine 요 숨기도스.
First three nucleotides are similar.
Same nucleotides.
Same,
three out,
yeah,
three out of
same.
4 are the same,
Yeah.
right? So 3 out of 4 nucleotides are identical. Are the same, good. Another group.
Ruka jenda.
아니 대 가서 대 코딩 베이스 라이저게. 티네 가서 G 다 CD 아니 RNA 다 DNA 니깔 치버리 샤스래.
When they pair together, G to
pairing,
C,
the pairing, right?
G and C,
Right, so you have
GC, C.
C pairing. Right? Maybe one more?
니가기 니가 냐다 지금 총벌이 샤. 니가기로 내준 냐다 셔주도. 어디 셔?
They store information.
They store information, right? So DNA and RNA can actually store information, genetic information.
Yeah.
Good, very good. How about differences?
다 개별가리도 우사. Yeah. That did one. One strand and
Yes.
double strand. Yeah.
Right, so RNA is single-stranded. I cannot spell. And DNA is what? Double-stranded, right. Good. Another distinction.
Yeah, sugar. Sugar is different. Sugar means
Sugar. What about the sugar?
Kari chepe, karisre.
vanilla deoxyribo deoxyribonuclease ribonuclease
Yeah.
Ribose and deoxyribose.
Yeah.
ribo and nuclease.
So chemical differences, right?
Yeah.
Chemical structures are different. Yeah. Good. What else?
Base pair differences.
La.
Base pair differences.
Best pair.
One is thymine and one is uracil.
How are they? Yes. So RNA has uracil instead of what? What does it replace in DNA?
Uracil tiki jenda kari chika chapchi yoresa.
Thymine.
Right. So uracil in RNA, thymine in DNA, right? So the pairing is actually different between RNA and DNA.
아니 질레 채워야 이렇게 질레 RNA 있는 그 프로틴 튜버 지기래서 아니 DNA 게 지금 말해서 돼.
The function are different. RNA produces protein, DNA doesn't.
Yes.
In that
so
stage.
their functions, the
Yeah.
way that they function is quite different. Yes.
Function.
And this becomes a very important issue when it comes to the central dogma. What can RNA do that DNA cannot do when
that
it comes to processes.
The chele tang juram go gane keche she bai bai na ane tanda nga chu tawa nyong pol he pika sa ta ke chambu chag yores garchi la bai na ane DNA gi chi mathia RNA chi thka kari yores te na ba keche she on stambala ne tanda gi chele ke chambu chag yores.
So I'll ask Dogma when we go on break to translate this, so you can take a picture, so you have it for your notes, for your learning and understanding.
Yeah.
Okay. Thank you.
Tada the person barone pegina.
So one group mentioned, I'm just going to do it by hand. One group mentioned that there is a difference in the chemical structure between DNA and RNA. So by understanding the differences, right, you have a carbon ring in both RNA and DNA, right? Where is a difference between them, right? Which one has what? So talk about it for about a minute, and then we'll see where you think the differences lie between RNA and DNA.
तो तंदर दिन से जिके कल्चर से संभलाया DNA तारे ने ये जी कोई बच्ची और वाह तेला या जी कोई बतले खेपे और इसे गोरेस तंगा सो DNA तारे ने निश्चय ना या अने गसु कार्बन के along to or what rinse growth along the ures go good to your along the ures the tiganane kogi ta koga nisa kolas through chashi kolaya and a koran zegi keperti drugues and a kamachis knalaya to the churches.
So we have RNA, DNA, carbon ring, carbon 1, 2, 3, 4, right? Carbon 1, 2, 3, 4. But there is a difference in carbon 2 between RNA and DNA. Think about what would go in RNA and DNA. Talk about it and we'll see what you get after we discuss it.
DNA pajote the RNA sejote bana DNA ro DNA na la ya ngasugi carbon galon to yo bache carbon ge alon yo pakansilane na carbon ge the tangkat sajute carbon ge antang terwa carbon tampon jo sambajo se gorwa ta tanden din na khona cherpet rosa chote ane kasa carbon yo ge te sabla du gores ta de la ya cherpet kari ores ta de la kari chagu yores te kamachis na la to the chiroches.
So discuss it for about a minute and then we'll see what everybody thinks.
Kamachis to the churches.
No, of course. They have their answers in their PowerPoint.
Yeah.
Everybody went through this? Good. All right, can anyone tell us what you think goes on the carbon 2 on RNA?
RNA carbon yogi dela ya zikari dugo sares.
OH. Very good. And what goes on the carbon 2 on DNA?
Dela?
H, hydrogen, right? So that tiny little distinction between the hydrogen and the hydroxyl between RNA and DNA makes a world of a difference in their ability to process and function.
Aha, ta tanda dela ya ko phajo tela ya OH la ro de RNA la ra ani DNA la ya H chipro wa ta tanda dela G chani ke che shebana khapti simashme do re bare yimbai na ya tela tende de te ko gi chele gi chala ta pe chepe shu chu zu sha yores.
So between RNA and DNA, which of the two is a larger molecule?
Aha, ta ga su dus ta bai bana swa RNA ta DNA ta bai bana korange didir wada didig chane didig kangi chiwa yosares.
DNA. Why? Because it's double-stranded, right? RNA single-stranded, DNA double-stranded.
Aha, ta de ko ge de karsan didig chane she bai bana DNA chiwa yor koran yang mani yor wa RNA yang ma chima chiwa mara.
Here's another question. Which of the two is more stable, RNA or DNA?
Aha, ta twa chi yores ro DNA ta RNA drubai mana kangi chi temba yores.
DNA, right. And here is a really hard question. Which of the two is more versatile in their function, RNA or DNA?
Aha, ta de la twa khabuch tang yes ro ane DNA ta RNA khurangi jele gi chane she pai bana versatile system bala ye thupu se gro da ko new ashu chambu do se gro wa stembu do da tena do ko or te kangila ya new ti ores.
RNA, right? What is it that RNA can do that DNA cannot do?
Aha, ta de twa yang se chi yes ro DNA gi dim thu pa RNA gi dru thu pa ka ore sa. Kalchi yore ende lai de ko ende ha
RNA danda nucleus nalo ne shlo ma ten ya this protein so to DNA nalo hat nucleus nalo.
Aha. What Camden says, DNA stays inside the nucleus. RNA can go outside of the
Right.
nucleus.
And what is the importance? It can go out in the process of replication. Something that DNA, even though it's far more stable and lasts a lot longer, it cannot perform replication. Only RNA can perform replication.
Aha, the replication system bala ga sun dashu zuya chigoro and dashu zuya derwa. Ta RNA ko ge chlo kanda stong gore system ba dashu zubala tani chongo yores ro. Ani dashu zuwa tani chongo yores. Ta de kapsang aso DNA te ko ta tembu ta yore bet tembu yor da imbaina ko ge dashu zuya ge korange chelite dapt kamares DNA ge.
So a really important and truly wonderful idea is that such a small difference leads to a complete distinction in the ability to reproduce.
Ta tanda de bana digi ro koran shala ya zigi chane bani chebti simashiyo mares. Imbana ta dela tene de ko ge ta reproduce the sambala chipe chiagi nubatila ya ani koran cha chepe shuch yongo yores.
Good. Any questions?
Draw ayesa.
Good. All right. So let's go through the steps of the central dogma. And this is working now, so it should go pretty smoothly. We go from DNA. What is next?
Aha, ta ga chu ge tawa nimpu ge kurum chioro wa pangrum chioro te ta ge ins ta tampo DNA res. Ta DNA ki chila kal chigres.
RNA. Good. Good. RNA.
Aha.
What's next?
Tigi chila.
Of course, I don't understand it, but the names are there, right? Silly me. Protein, and what is next?
Tigi chila gare.
A trait, hair color, height, skin color, eye coloration, anything, right? So you have, yeah.
Tigi chila ya cheshi ting goris, cheshi ting atsu ge chiga kaso gore zupu ge rang se tagen do wa ane papa gen do wa miki khatola so batigi cheshi ting goris.
So you have the manifestation of a physical property within an organism. And that could be anything regarding a physical property within an organism.
the cheshi sigasa gaso the chengwe chike ne zupula yopi the chechu karina kunat kores.
So the important thing to remember is not only the flow between DNA, RNA, protein, and trait, but what we call each segment of the process.
DNA jila RNA tijila tijiji tijila cheshila ndubi pangram tingyo tsa maimba te gala karinga haku yori system balaya re ne dhua gi jurum chiorwa te ngi gugores.
So we go from DNA to RNA, we call it transcription.
Da DNA ne RNA la trip pi rumba tila ka sigres na phapshu sigres.
What do we call it? We go from RNA to protein.
RNA protein la dhua tila ka lag raha.
Translation, right? Oh, by the way, before we go into translation, transcription occurs where in the cell? Within the nucleus.
Tatanga su phapju saiti kecha mashing la ya tu staba bana phapshu saiti kawa chungure sana phapshu saiti thapongi tinying ginala roa tinying ginala chungores.
Right. So we have, you could envision here is a part of a cell, the segment of a cell. This is inside the nucleus. You have your double-stranded DNA. It opens up, and you have an enzyme called RNA polymerase, which produces an RNA product. This is transcription.
Tinying nalo la chungu yores ta DNA kharchi gore sena DNA nyama ni tigna ne ta nyama chi chagu yorwa re mare nyama chi chaya la ya ku cha ba chi ate la ya tizi chi yores ta tizi tig mele kar re sena ane karsa RNA polymerase tanda dinga su ge mangjor menji chi yorwa mangjor menji ti sigres ta tege ku nyama chi zu de te ane ga su ge phapsi gi juram ti yong chi yores.
Right. So you have your double-stranded DNA. You have this polymerase. Right, the DNA polymerase is an enzyme, and the product coming from transcription is the RNA product, and you know this is RNA because we have uracil instead of thymine.
Dada de la taba bana tanda sarpunde nga su manjur menji tir wara ne manjur menji tin bana ti la tene de ti karchi gore sana ane ka DNA ni RNA la phapsu chi gore sta phapsu chi bate nga su khoge ta DNA la nyama ni yor wara mare nyama ni la tene de ta nyama chi te phapsu gore wa ta dada nga su o kite RNA imba ha kures karchi la pa bana nga su khorangi nyamdu trichala ta pa bana kare du khoga la ya uracil duwa uracil stambala de ne nga su RNA bahakures.
And what is important to note is the way the RNA is produced. It is a complement of the DNA strand.
Da RNA ta DNA kari hakure system bala ko phapsi chibi kapsa ane karsa RNA ti kana do chi tukku yo metila ta ke chambres kachila bana RNA ki tupi kapsu khurang gi nyangdup tcha re te ane khurang chi tambo DNA na ne khat dikas gore ko chat ge gi chi ro khakon che ge chat ge gi chi ro wa ti nangje ane chat ti ta ya te ko ta chat gi chi yong yo res tabiate.
So as the polymerase moves from the 5' end of the DNA to the 3', it's going to start complementing the DNA. So for example, we have TAT. The complement would be A, that's a complement of T. T, which is RNA U, so it's AU, and then TA.
Tanda de bana ko jusa de chujok chala dene yong bache ane ko Kurang phapsi chibi kapsa tanga su cha dugu yorwa re cha dugu ti nyangdu ge cha dugu yorwa da cha de khakong se gre khakong wa chigres da yong de kanda se T na A che rwa ni shende de A ji la ya U che ani T ji la A ti yong gores.
So the RNA is a full complement, a mirror image complement of the DNA strand, the coding strand.
Da te ge korang ge nyagmati ka na phasuk ke khakung ka na jeng ula yong yores.
Okay? And all this happens, the process of transcription creating the RNA product always happens inside of the nucleus.
Da te tanda ga su ge phapsu chia ge rambate katu imba na ya tinying gi ro thapung tinying ge na lo la chung res.
Okay? Are there any questions?
Try yes.
Good? All right. So when we go to translation, which is the process of going from RNA to protein, we call it translation, right? We go from RNA to protein. Where does that happen? Outside of the nucleus.
That tige ramba jibadi karla na RNA ne tiji tripik ramba tila nga chuko phabjir se grace phabjir koala chungore se na ne tinyingi chlola chungores.
So the messenger RNA that is being created in the process of transcription travels outside of the nucleus through a channel. It enters the cytosol, which is the cytoplasm, the medium of the cell, and it attaches to a ribosome.
Tanda nga chuki tapungi tinyingi tripiki phabjishibiki ane ti pangchin RNA chiwarwa ane ku julam chikje tinyingi chalokla thun ane dila cytosol sikdo cytosol sede nga chuki ti karsa tapungi tatsi ora tatsi ki chashi chigres da tene ku ti juchi madu te ku koala ya jargre sena ane ribotazila jargyo res.
And what happens here, when it attaches to the ribosome, here we have a close-up of the ribosome. You have the messenger RNA coming from the transcription outside of the nucleus, attaches to the ribosome, and again it's going to complement the coding of the sequence, and the ribosome starts producing protein, a chain of protein.
Delaya ta dinga su di ribot haziti ku this cher tang bai bana phajo tina do chingong yo reswa na te gang la nga chu pangchen RNA te dos dwarwa ne ku gi da chi gorwa sangda te re ola dwa ta ribot haziki ku sangda re te sangda te tak peshe tang de ta ne ku tzi zu res ne tzi gi ta chain of system balaya tzi gi thama se gro ta tzi mangbu du zu res.
So in essence, we are changing the language in the process, because in transcription, we go from DNA to RNA, which are still very similar to each other. We're still dealing with nucleotides.
Tanga su di kas phabjit chi bi kap se yang khuga dalarwa dalan jwa tang go res ta ku so da chilen pha de ti ge da ge yi tilan jwa tang go res tanga su phabshi chi bi kap se ba na ga su DNA ge da ti RNA ge dala jur gu yor da imbai na ya ti kap se this the dachwa yo res ka ti kap se ga su ti kari pe tang go res sta bala ya ti pe tang go ro nyang de tup cha ti pe tang go ra te tang go sang ko this chando yo res.
But now within the ribosome, we change the language, because we have our RNA product from transcription matching with what we call an anticodon within the ribosome. And based on this matching with the anticodon and the messenger RNA, we produce amino acids, which are the building blocks of protein.
Pangchen RNA ge gangla yo pi ge ta nga su this codon system bala nga su ge ta sangda gi chemba tirwa da gi chemba te ta thumbi gi ribosome gangla kariore system bala anticodon da ko ge dogda se go mro da tina do chi ore swa sangda te ge dogpa ta khu ta thuka jan chi ore ta tela tere de te ane nga su kari chi ge system bala ya ane ti ka sa ane tji gi ngo de zu yung go res.
Good. So in transcription, we use nucleotides. We go from DNA to RNA. They both use nucleotides. In translation, we go from nucleotide to amino acids, proteins.
DNA ni RNA chi because phabshi chi go re ti ka sang asu wa nyang du tcha ti peche tang yo res ra ani ti je la ya RNA ni tizila typical sang asu ge ta phabji chi ge ramba te la ya ani kare peche tang go res na nga su amino acids yo ra amino acids peche tang go res.
And as you will learn, as the ribosomes start creating the chain of amino acids, which we call a polypeptide chain, this will give us specific proteins with specific properties.
Chesugi kale thongres tanaga surabo tazuki ani dela ya titsigi thawach du staku chetanchi ngorwa titsigi thawach chetachi batigi ta susu gi chechu mandawala tendete ngasula ya tiziyang mando ngores ti tanaga su kale thongres.
Are there any questions?
Good.
All right, so here is what RNA and DNA look like in an actual cell, right? This is how you could actually visualize it if you were to look at it under a microscope.
aha ta ga su ge de na la ka sa chi she go la ta pa ba na ta RNA ta DNA kan da do ji ge pa te ta chi deng na na ni cha tong chi she on ta pa ba na de na do thong gres.
So you could imagine you have your line of DNA, you go to transcription, and you have here all the little segments of RNA in the process of transcription.
aha ta de la ya chi su ge DNA do span ge yo ba che ti na ne ta da rigit sha ju sam de la ya ko chi ge na de ju do chi do ga ta ti ka la na ga su ge phap shi la te ni de ti ane RNA tripi gi ta ko RNA gi ta yan lak se gro ta ko ning la te da yan lak ma RNA chin chin te na do thong ge yo ba de res.
And of course, the RNA would eventually break away, leave the nucleus, attach to a ribosome in the cytoplasm, and be translated into unique specific proteins with unique specific properties for expression.
aha ta da ta rim shin sro ta ga su tin yang la yo pi ga RNA te chu te tin yin ne chlo the ko robot hazilan jer de te ani ko che chi mu se ba ton dem bi ge ani te zi de du che tin chi yo res.
So you could imagine all those RNA polymerases, all those enzymes moving across the DNA sequence, producing nascent or newly made RNA product, ready to be translated for expression.
aha ta da de ko ta de wa ga su ge ning ko te ru do ning ko ma ra ning ko ju chi ma thön du ni ga RNA te zu ge ta te ta ke ani ko ga phap zu thuma te du sa zu ge yo res.
Okay? So we have a little activity. This will take a couple of minutes, won't take too long. We have a couple of questions here. We have a drawing, basically showing a double-stranded DNA. You have the priming direction. You have the presence of your RNA polymerase and the product of your RNA.
aha ta da de che su che gu chi yes de dry yes ta de na ta ba ba na ga su ge di ka sa DNA nyang ma ni che ani ko ge ta ko tha ni te do da carbon ko yo me te do da te na ne ga su la ya pang chen RNA chi ba che ne te ga la ya yi ge tu sha ju de ko rang ga ta sang da ter wa sang chi ter wa te res.
And if I was to give you a test for this messenger RNA and ask you to label the prime directions of your messenger product, where would the primes go? What numbers would go on each side?
aha ta dan the prime say de ko ga chang ka te ta tak ti ro da tak ti dan da de le sum tang ga ta sha do te ke ting gi chi su la tra tri de ti ani dan da pang chen RNA ge ne ge ta chang ka ti ka su re sta sum tang ga ti sho ko la ya sum re sho ko la nga re ti ke che tri ba ba ne ti kang ge res.
So would the 5' go here or here? What do you think?
aha ta chang ka nga yo sa ge ne chi ro wa te ma wo te ro ya gang te res.
Anyone want to come up and point where would the
Yes.
5' go?
Yeah, they say in the lower one, low range.
Here, right? Absolutely. Why? Because it's a complement of the template, right? And a complete identical version of the non-coding strand.
aha da te dan da go de cha go yo res ka shi la ba ba na ga su ge ko ta ka su ge phap shi chi bi ge kap sa ani ko rang ga phap shi chi sa ge nya ma te tang kha kung ga rang jen res te ta cha de res ro ani phap shi chi sa maim bi ge DNA ge nya ma jen ba te ta chi pa chi song res go ro den sta ba la ya nga te o de la le ko res ta da ko ge nya ma o ma te tang kha kung ga rang jen che kha kung ngo yo pa che
And in which direction would you say the RNA polymerase is moving, from left to right or right to left? Which of the two?
Ta de manjor manji chora RNA manjor manji de ani yone yela dugurwe yine yona dugures.
Anybody? Raise your hand if you think it's left to right.
Aha yone yela dustena yone yela dunjire sabana lapacharo ches.
Raise your hand. How about right to left?
Yine yona dunjire sena lapacharo ches.
Let's see who's right. Come on. It goes from left to right, moving towards the the 5 prime of the non-coding strand.
Ta tanda di mana ko dueticare system balaya ko dichigorwa ta nyakma dichigorwa digi ta anga ngayo sachtoa tigi shola dugres.
And if you were to label the strands, which of the DNA strands would be the template or antisense strand?
Ta tanda di nane chesuge ta ba imbana DNA gi ta nyakma yorwa ta di nane template la nga su mapi gi nyakma chigorwa ta mapi gi nyakma ti kangres template te.
Is this the template or is this the template? The top one or the bottom one? Raise your hand if you think it's the top. Raise your hand if you think it's the bottom. Not everybody participated. Everybody participate. If you think the top, raise your hand. We got a few people. Bottom? So it's about equal. Let's see who's right. Template, antisense, is the bottom, which is used to produce the RNA.
Template lana pishir kwa tede ta zugu yorwa tanda digi pishigi nyagamati ana tika res na omati res.
The strand going from the 5 prime to 3 prime is called the coding for a very important reason, because it's identical to the RNA product.
Ta tanda dirwa ta gangatila ya ane ngasuge dadi nyagmase res te yang ko minti takate ke chembu chago yores karchila baina tanda ge nyagmati wa dadi ke nyagmati ane RNA ge nyagmata ta kasa ta chachon res this dawo yores.
So the strand which complements itself to create the messenger RNA is called the template or antisense, and the other strand is called the coding or the sense strand.
Ta tanda dege ko DNA ge nyagmani yorwa DNA nyagmani yopi ge nane ani ko RNA ta tbyala shi shasa chiwarwa tela yang asu peshi ta peshi nyagmata yana ta de sense de ko ku na hakomas ta chowa do ku bana ne chowa ta dokchor su gum ta antisense na dokcho ge nyagmas gres ro ani gang tela ya ta ti maim ba gi senda tela yang asu ge dadi nyagmata yana chowi ge nyagmas gres.
And it's very important to understand the direction of the movement of RNA polymerase, which always moves towards the 5 prime of the template.
That dig jail, I just carrying a Google Senangasuge RNA manjo menzich or a dig go Guchetan Saga Kashoting Ugures, Kukare Sena, Natsugu Pishigi Nyama Tege, and the Angangayo Sagi Nemu Shola Katim Bana Guchetan Grace.
Good. We good? No questions? Excellent. So we know this is transcription. We're making messenger RNA. We're dealing with nucleotides. Same language.
That other day, Kapsaka, that's a public Keshekora, public Keshekore, the Chongade Kawachongore, Tining Nana Chongore, and the Korange, Da, Keta Pechetanga, Kari Pechetangore, and the Kuranga Daisigro, that the Igiti Peche Karitangores, and the Yanda Tripcheti Pechetangores.
But now, we're going to change the language.
That other Karchigas and that Natsugeta, Kate Segurata Igiti, and Jordan Gains.
So when we go from transcription, we go from DNA to RNA. In translation, we use triplets, a set of 3 nucleotides, which all codify a different amino acid.
Tamputela so DNA ni RNA la ya phapsi ka simba na ga su ge ta ko nyangde tripchati pe jitang de ko datima ko tugu yorwa ta tene ga su phapji chi ka simba na ane pancha RNA na yo pi ge ta ko ge dai simdi chamba chi ju de ane ko karsa tji amino acid chi yorwa amino acid manda dus chagi yores.
So making the jump in translation from RNA to protein, we no longer use nucleotides, we use a set of amino acids, which is a very different type of language.
Tanda nga so karsa dine pancha RNA tjila yang ko phapji chibi ge kapsa da karpeche tangre sena ro ta data kiti karpeche tangre sena amino chirji gi ro amino chirji gi ta ko nyam te debala te ne de te ane dati pechu tang re ti stambala ya ti ane gala nga su pechu tang ai ga da chi ro nyang de tripcha chi ro ti ta beti lo res.
And this becomes important because we have a potential of 4 distinct nucleotides, but we only use 3 to create each amino acid in our polypeptide chain.
Da de kapse yang ke chambu chago yores ta de la ya ko chepra shi chambu chi yores ta nga su la ya DNA ni RNA la ma phasu chong sta bala nga su ge ta nyang de tripche te ku ma do shi special ta yang go wa re ma re yimbai na ya nga su tzi la ma phalum chong sta bala ya shicha nyam te dra de chi go ma re ku sim chi go sim sen te de peche ta yang go re sta de ke chambu res ne ta de.
And this is important because by using only 3, we create a level of redundancy for conservation of sequences.
Handa di karichin ke chambu chago res na sumdi chi de te ne nga su ge ko ge chi khörim ti nyar sa chi ge ne tang de ngo res.
So understanding that each set of 3 and the order becomes important, they create different amino acids. Let's take a look at how this process of translation occurs.
Handa danda de kapse ko panchi RNA ge sanghi chi gorwa te sumdi ma yong do re mare sum chi de te de yong de kapse ko sum UP ge igi sum de ge yang pangram te yang ke chambu chag yores that is damba danda nga su ge de ko panchi RNA ane ko tzila phapsi chi rba de ta was.
Right, so we have our amino acid chart. And we'll spend a bit of time going through this because it is a little bit complicated, but we'll make sure that we all understand it.
Dada de la yang aso amino chirji gi rimu chi yores ro dus ta ba na chu nyukta chaura chi dus ne ba na ga su ge kale de shi ja chi ngi ba chi was.
I actually created this by hand, believe it or not. I drew it and I put all the names. And when I was making this, my wife came up to me and she said, "Are you making a game?" And I said, no, I'm working with science.
Ti mana gela ge lakpa gita dus remote che dus zo ba in standard remote ro this zo be kapsa ne ko ge chime yong de ale chorong ge simu sia chi zo go yes dusa ne mare simu sia chi mare de chari tan tham be ge ko ge chi ne ta chu ko de shi ja go la ba yes.
Good. So how does it work? And you may see different versions of this. You may see charts that are square or even different versions that are also circular. In my opinion, and you may feel differently, in my opinion, the ones that are circular that go outwards are a little bit easier to read and apply.
Da chesuge the amino chirji gi remove de chesuga namba mando mabu thong yo gires chi china khashi gi dushi thong da khashi gogo thong damra thong yo gires ro da ngai baina dine do zo de tang ta ko loya ti ro kimani thala dus loyong be kap search gi loya tab du chi dus.
And for your activity on the second half after our break, you will get to use your own chart to get that hidden message in our central dogma.
Da nga su person ge je la ya chesu chegu chi drigo re chegu de dribi kapse tanda chesu ge de ge rimu chi do ga de pe che tang de de ane nga su tawi nyimbu na la yo pe tawi nyimbu pe de pe che tang ani tawi nyimbu de je de te ne ko la ya sangda chi kare chi do de troba chi gures.
Okay? So let's apply, let's learn how this works. You see 3 different layers of nucleotides.
Da de chi ta ba ba na nga su ge nyang de ge tricha inane ko ramba sim chi do ga du sta ba ina.
Okay? So you always go from the middle outwards.
Da ko lo be kapsa kana slokores na kimani thala du slokores.
The middle has 4 quadrats and you get to choose one amino acid, either G, U, A, or C.
Dat chima to ane kar chigures na chorong ge yangde tricha kang chi dam gures ta tigna na ga sur G U C A do washing na chi dam gures.
Okay? So once you choose your first amino acid in the first level, you go to the second level. And an easy way to understand it is that the first level has the largest letters.
Da ting chi kuala ta che te che ta rumba tangpu te na chasna rumba nyo te gures ta rumba ti ya chi ko lil hau cha ba na ane ko rumba tangpu te na na pi gi te ta ku ye chenpu chen do chi shi chi yes.
So you choose your first letter. Let's say we choose, anybody give me a letter. Choose your letter. What do you want me
Chi
to
damro
use?
chi da sa ye gi tangpu te.
U. Let's do U. I heard a lot of U's. This is the first letter, right?
This
So from here, we'll go to the second section. which is the letters that are mid-size, not super small, not too large. This is the second section, okay? So we chose U, so this is the second section for you.
da tangpude nga su igi tangpude yu dambarwa. Da digijila rumba nyote dangurwa. Rumba nyote ane ka igi yang chi chenpo mayamba chonshamba nono chidwa. Da digi nani kula yu la ya kuta nyamte duwagi la ya igi shi chidwa. O te nani chidamgures.
So give me the second letter. We have U, what's next? A, okay. So we go UA, that's the second section, right? And now we go to the third section, which is right here, with the smallest letters.
Da nga su yu dambara tangpo nani nyote nani e dam songa. Da digi e gi thala ya karedo da nga su ka rumba sumbate dugurwa. Sumbate do na yang kula ya igi nyi cho shidwa.
And this is, my friends, where you see the redundancy. You go UA, and then you have another option, right? You can go UAU or UAC.
Da dila ya ta chi karsa chi mando ta chi cancer ga nang na chi yores. Ta de gare se na da de kapsa chu dem ba na UJ AJ tigi jila ya U da na la degree C da na degrees.
So we'll go to UAU or UAC. Each option gives us tyrosine, the same amino acid.
Da nga su UAU in a degree, UAC in a degree ko kangi imbai na tila tende deta nga su la ya amino jurjiti kangi degree se na tyrosine se de degrees.
Likewise, if we go UAA or UAG, we have what? Stop. So the protein stops. It is no longer translated.
Tada nga su ta UAA re UAG imbai na tila tende deta nga su la amino jurji karyong gore se na dati karyong gore se na dati 참샤 사이티 다티옹고 레스틴 스타발라야 네 드지 다 드비아기 린참티샤고요 레스텔 참샤 그레스.
And that, my friends, shows the order of the triplets. You go from the beginning, the largest section, you go outwards, depending on which one you choose. Let's say we choose G, okay? We go GA, and then you have other options. GAA gives us glutamic acid.
Tanda nga suga loktang research ja songro ada ka jurne ke tiwani ya dugo resta pisha shana nga sugi tiwa tila tangpo tila igi chishu ti ji chiti jile e chiti jile yang e damba imba na nga sula rati karla resna glutamic acid dragures.
Anybody want to come up to teach us how to find your amino acid that you want to find?
Europe?
Anybody want to come up to do one for us? Yeah, anybody want to come up?
Okay. Tanda chechu na ne chi ya phe de den den nga sula ya amino chirji chi the kanda sego yo me te chin de shi ja ro da sa. Sure. Da sure.
Let's not all jump at once.
Ya chama sho sa. Simon, ya chi sho.
I'm going to pick somebody.
Oh, yes, that's good.
Good. All right. Round of applause. So which one are you going to do, my friend? A? AU? So there is redundancy, right? There is redundancy. If you go AUG, what do we get? Methionine, right? The other three options, AUA, AUC, or AUU, we get, right. You see how this works? It's redundant, right? Good job. Thank you. Now, in some cases, In some cases, we do not have redundancy. So for example, tryptophan. There is only one option for tryptophan, which is what? U, G, G. Right? So only U, G, G, only that option gives us tryptophan. So some amino acids have redundancy, some do not.
Handa dada de kaso imbana ngaso ge picha shana dela ya tryptophan says to dida do imbana ko ge UGG ro ko ta ko ge sangdate kanshik mashi ko tege yo marwa kashi chi imbana ko sangda ni sumi shi tege do re mare dis tambala ya kashi ya gunyok pore dita imbana kanshik lila u tse yo res.
And beyond understanding the coding and how this works and how you build your polypeptide chain, there are 4 sequences that are very important.
That and the kandas that the kandas jungle yomete hakunu jelaya and shenda yang asuge ko kas kurim shichi or wakurim shiti and kechambuchagres.
Anytime translation begins, it always starts with what?
That andanga ani projecting good to be capsules katimana karili tampon good to yores.
It always starts with methionine. Always. The beginning of a polypeptide chain always starts with methionine.
The projective capsule as to tangwa kerwa tangwa chike Gunzixati Katu by Nakare Sena methionine inside there is.
So AUG methionine is known as the start codon. This is very important. Okay?
That under the AUG doa AUG dig that sound like Sambadelaya Negasuga code that to be caps Netsambadi the Gunzuki that sound as a goris.
You can also have a polypeptide with methionine in the middle. That's okay. But the beginning of every single polypeptide chain always has to start with methionine.
That is that the during becomes an interested Hawashika parola methionine said the young at the use baris, Youngres, Yugres, Tinado Neton Youngres. Even at that under the capsing equate, good to pick up, sir. Carriers, Katina.
So methionine functions as both an amino acid and the start codon. It has 2 functions. So we have 1 start codon. What else do we need to end the peptide chain?
Tangasulum good Chandadera, Chandadera, Tangasuge, Koge, Tetsit watching Jigdom Guyo Pina, Delangasula Karagores, Chanda Kangores.
We need to stop it, right? Good. Looking at our chart, how many stops do we have? How many stops can we have? I'll give you 30 seconds. Look at it. Talk to your groups, 30 seconds. How many stops do we have? Talk about it. Talk about it.
Stop Katsudos, Ruatella, Judomete Katsu, Taro Chesa.
So we have, raise your hands with the numbers. How many do you think we have? Raise your hands with the number. 3, 3, 3, 3, 3, right? 3 is a number. So we have 3 potentials. We have excellent. So like I tell my students, I won't make you memorize them. I do make my students memorize the 4 codons, the 1 start and the 3 stops. Maybe I'll make you memorize them as well. I don't know, we'll see.
That's good. Palaya Koloptulaya teaching yes ro da ko rizig changda shi ngachur tang chi yes ro karla na jigdom teacher makar sang gun zuya ge rizig changda chikche ani jigdom ya ro chamche ge jigdom ya ge rizig changda sum do wa shi de ngachur tang chi yes chesula tang ma chi guna chi chi na chesula ya de ngachur tang chi gu yes re sa.
So very important. We have one start, which is universal, and 3 different stop possibilities.
Anda deke chumbu chago res tela yang asu rizi changda gi nane gun zgne chiche ane jigdom ne sandoa.
Any questions about using the chart?
Tanda ge remove the pechu tanga gi thela ya twa yo sa.
So after we end our class, even after we leave, after the end of the week, you will have your copy of the chart, practice with it. Always make sure that you know how to use it. Because in your future classes, when you teach this, you have to be able to tell students how to find and how to create peptide chains based on the triplets.
Handa dada nga su zendra de chanare chingu chane kan zendra sa ba ina da dendra chi dawa chi karim ba na che su ge tanda gi remove the pechu ta ya la ya ane su su jongda chi ro chi sta jema chi su zendra la ya trions ta ba la ya ane de pechu ta de ane nga su ge tsi ge changwa de zo wi kap se ane de kanda yong gu yo re sa de de shi ja bi ka se chi su ge de pen to bu shi yo re de sta ba la ya ane jongda chi chi su su ge ngi wa na ro chi.
This took me about 4 to 5 hours to create, but I'm very happy to give it to you that you can practice with it and use it in your classes.
Handa de zuela ngara se ba bana chu shine nga de gor sons ko zuela man tang I de chesula ya chiga chesu ge chesu pecho ta ya ta ne chesu ge jiman zendrat tv ka su pecho ta che de dus khotro che de ne chiga gatu nga ne khotro chi yes.
Good. So beyond understanding how to create our chains of amino acids, we have to also understand how different amino acids react in the environment.
Handa tanda tampote nga su ge remuti ju de de nga su amino chirji de zotang de kar su de ha ku nu jela ya ane amino chirji de ko mando de ko khori mando na ne ko ge da jinjur sana ko ge jeli kandas tenge me de ya ke chemu chag yores.
So we have our set of amino acids which we can use to make our proteins. And by the way, the potential of different amino acids is far larger than the ones that we use in life.
Handa danda nga su ge da chi nga su ngoyo ge su na la pechu ta ya ge amino chirji chi gorwa de la temba na chi ta yu sipi ge amino chirji khatang te dele chi ma yores.
Cells use a total of 20 different amino acids in life.
Handa danda nga su ge thapong ge bana ko ge da chui rin la ya su ko da ko ge amino chirji ring mando nish thamba de pechu tang res.
You could potentially have hundreds and thousands of different amino acids, but only 20 are used by our cells.
Ta chorong la yu danda amino chirji ti pe te kura tonta mabu tenda yu chopa res bana nga su ge zupu nga thapong ge amino chirji ane nish thamba pechu tang res.
And we don't really get into this in our class, but I'll mention it. Based on their chemical properties, on all the different amino acids, they're going to have different reactions, and they're going to interact differently within cells.
Da ko la ya zegi taptang theni ko la ya chechu mando doa chechu mando yo batela tene ko thapong na la nebe kapsa thapong ta nyam ta ko jijon duare cheli duare de ge chi number te ko mando yores.
So for example, some that have nitrogen are non-polar. They do not have a charge.
Da tanda picha shabana tanda dela yo bigite ko nitrogen yo such doa de bana ko de anitang madam baris ni madam bain stambala ko ganglaya lokhurti yo maris.
For example, phenylalanine has a carbon ring. It's also non-polar. It will not react readily to water, for example.
Da tanda de phenylalanine such doa de bana ko la ya yete ani carbon ga alon chi yores tim se ko la ya ne ko la ya nitang madam baris tin stambala ya ko karsagorda telatene nitang dembalatene deju chunga gi netse de chunga maris.
You may have an amino acid with a long side chain which gives it a charge.
Da tanda ya amino juzi khashim bana kola ya sula yo pi thangwa te rengbu yo sa tena dwa Istanbul kola ya yang ko khutang dang yo res lokri yo res.
For example, aspartate has oxygen. Oxygen has a very strong charge.
Da tanda de la ya aspartate la sidwa tanda shiba de gang de la bana ko ge thala ya de cholong dwa cholong em bana cholong la ya ko ge
And that's all I'm going to say about this. In chemistry, you may learn that the different structures are going to give a different charge, either positive, negative, or even no charge. And that will dictate how the amino acids will interact within the protein.
Zinjur pagangle talaba imbaina amino chirji su lagi chechula tene dete khashila imbaina ko lokriyo ba khashila lokr me ba khashila ya ane kasa khashila ya lokr te phutang da ba khashila lokr motang da ba khashila lokr me ba tetla tene dete lokr yo me ge netan tela tene dete yo me ta yo pa te phutang mo imba ge netan la tene dete thapung na ne ko jeli ten ta phasula dejo chitante mando yo ba te ane zinjur pi kapse yang ko tajun de jagres.
Are there any questions?
Yes, sir.
There's a question. Yeah.
So I have two questions.
Yeah.
The first one is like, can mRNA stent make different proteins? That is
Yes.
the first question. And second question is, if our body, the cells in us, can make proteins, then why do we need to eat protein from food?
Right, that's a very good question. So, can messenger RNA make different proteins? And the answer is yes. Different segments can be used at different times by the ribosome to make a different type of protein, a different sequence of amino acid. So yes, you could have multiple proteins coming from the same strand of messenger RNA, depending on which segments are translated.
Hada da draw ni ta sa draw tang pati kare sana nga chu function RNA chi ji de ta ane tzi ti mando tinda ane jus barwe sutkurwe ro te ju thgre is ka chila pa na nga chu ge tzi te amino chi ti ma zu yong bi kapsim ba na function RNA ti go ni men ta ko ge tripcha kangi chi phaj ji ma chi la ne tang la tene de te ane amino chi ta te la tene de te nga chu ge tzi te yang mando chi tin chi gres.
And the second question is, why do we need to take, if we can make protein, why do we need to ingest more protein?
That one you better carry Stambalaya Natsuki Zupuna Zipe Mabuduas. The team by Nangatsu Kalaganalaya to Zio Kalakatsin Saguris.
And the answer is that this process of translation of making protein is very expensive energetically.
Tatangatsu Zupunane dinner doge, Tata Tizi Zuagi, Rambach the Wa, Zupunone Tizi Zuagi, Rambuch the Wa, Rambach the Wa, that they pete. So
we need a lot of energy for such process, and that energy comes by ATP, which we get from ingesting organic matter, organic things.
And
if we did not eat or ingest nutrients for energy, our cells would not be able to produce the proteins. Good. Anyone? Yes.
Some shist in the chickens over the Hasidic mother mendo, then the she over in a cause shiga, chiquachiquat down young cadenas.
In chicken song.
So the amino acid, some of amino acid they need different codons. Some only one set of codon they make one amino acid.
Yes.
Some of they have a different kind of codons that make one amino acids.
Yes.
Whether these kind of different amino codons that make different amino acid, yes. Whether this amino acid is the same or different.
No, so it's redundant, right? So if I go back to this picture, oh, I'm sorry. this picture, right? So you could have different combinations of nucleotides, of RNA nucleotides, making different amino acids. So different combinations can produce either the same or different amino acids, right? So it really does depend on the direction and the combination of the triplets, and they make specific amino acids.
다다 gelagi toate ko sarwa ngasuge khashi mbana ane amino chirji de chala ya rizgi ge changda chamba chigi digdus khashila mbana chamba mando ni sumchi mbana amino chirji de chiba chizugdus tate tizu kapso chipa yora yo maris ta amino chirji de wa tate stabala tanda gelati kas desija kores tamba ngaso tate chirge na khoge neta ter do ta in stabala ngaso de nyangdub go tcha samre chi de ko ane karso gore de nyangdub tcha samre la tende de ngaso amino chirji de ri tu ko yora ra mare ta de ko phaju chi stambala chamba ko ane phaju chi ma chi ta chi ko ne gu zu ma de la ra lu gore simba na thama tumi on stambala ya ane amino chirji de ko kare chapati chi pares.
So for example, there are some amino acids like valine, serine, or arginine that have a lot of different potentials, a lot of different combinations can produce them. You have GUG, UGA, UUC, UUU, all of them make valine, so it's very redundant. In some cases, only one combination, like methionine, can only produce methionine.
Tada valine, valine tau bana ko chiga ta phaju chi dripsala ya ko traptong mando de dua karsa shi ko ge date dikta shi gane kono trit ko yorwa bana danda de la ya methylene sa dim bana ko traptong chi gane na ba chi gama shi ot ko marwa.
Okay, so the level of redundancy really does depend on which amino acid we're building.
Tada de ko ge netande karala ra ri se na amino chirji kangi chi chetan chi go metela ra ri res.
Good. Anyone else?
Trojemba use.
Another question is that, so the different codons make the same exact copy of valine.
Yes, yes.
Even there's like G, U, G, T, U, A, G, U, U, and all.
They make the exact same amino acid, correct. Completely redundant, yes. Yes, good. When we talk about mutations, that shifts, and you could potentially create different amino acids in the presence of a mutation, which changes the sequence.
Tada de kapsa chipa chichang se ta ko traptang te mando de chi amino chirji ti chi la ya ko da diktang te mando shi gane chi tube ngorwa re mare ta tripatele thamatele amino chirji chi tripate ko traptang jisha kange gane tripa bana thamage de karsa chipa chichang res ta tanda de ngasu ge jela de kapsa ta res tanda de ninjur yorwa ta ninjur ge kapsa ta de ta kabla ane chik shiching kari yong mente ninjur ke sta res.
Good. All right, so this is a good time to stop for our break. We have a lot of information, so let's synthesize this, think about it, take a break, grab some tea. We'll come back and we'll deal with other issues with conservation, and then we'll jump into our activity for the rest of the day.
Tanda so je person tangin tig jela ya ane ngaso ya yong data ane chi ta te gi chatn ti nyer so chi we gi netan ti gola ngaso yang she de shi ja che ane tig jela ya ngaso ge je gu chi de ya yus de ne ta chotan ni ba gi jere ane nyang kong jab ti samala ngaso je gu ti de ya yes.
Good. So I'll see you all in 30 minutes. Enjoy your break.
Oh yeah, this such a person to knowledge